Development of the endothelial progenitor cells, also termed EPCs, starts in the bone marrow with a hemangioblast, a primitive embryonic cell of mesodermal origin. Hemangioblasts differentiate into angioblasts upon absorption of VEGF, a substance secreted from stromal cells that stimulates new blood vessel formation. More VEGF and FGF originating from surrounding osteoblasts, the primordial bone cells, are secreted, turning angioblasts into bone marrow EPCs. These EPCs will further be affected by surrounding growth factors such as VEGF, FGF, GM-CSF and ANG1, leading to the formation of circulating EPCs, leaving the bone marrow into circulation. There are 3 stages to the development of the EPCs, from bone marrow to vascular endothelium, mobilization, migration and incorporation. Mobilization is the process by which the EPCs are activated from the inactive stage to the proliferative stage. Migration is the movement of EPCs through a distance, guided by the release of growth factors and other chemokines and cytokines. Incorporation is the union of circulating EPCs with the endothelium lining the vessel wall, resulting in vascular repair as well as angiogenesis, the process of vascularization of a tissue involving the development of new blood vessels. Build-up of plaque obstructs the blood flow in the artery, prohibiting the heart to pump blood efficiently. Lack of sufficient blood and nutrients in the region will lead to cell death. EPCs arriving to the ischemic area following the release of chemoattractive compounds, they then migrate through the vessel wall into the ischemic tissue and then release enzymes which will dissolve tiny holes in the basement membrane. The endothelial cells begin to divide and they migrate out through the dissolved holes of the existing vessel. Sprouting endothelial cells would then roll up to form a blood vessel tube. Finally, newly formed blood vessel tubes are stabilized by specialized muscle cells and blood flow then begins. This whole process is called angiogenesis. Cardiac catheterization is a procedure in which a doctor inserts a thin plastic tube or a catheter into the femoral artery in the leg. From there, the catheter can be advanced into the coronary heart arteries through the aorta, the largest single blood vessel in the body. The catheter is inserted into the coronary vessel until it reaches the area of vessel occlusion. During the procedure of EPC injection, after catheterization, a balloon is inflated and thus a small pool is created. The syringe containing EPCs is connected to the catheter and the cells are injected into the pool, which is in the area suffering from decreased blood supply caused by occluded blood vessels. Endothelial progenitor cells possess the ability to differentiate into endothelium, the layer of cells involved in both neovascularization, the forming of blood vessels, and endothelialization, the lining of their lumen. These functions of the EPCs enable the development of new therapies that aim to use these cells for the treatment of severe cardiac disorders caused by inadequate blood circulation.